In this video, we will discuss about Chikungunya virus that is caused by the mosquito bite. So we will discuss how this virus gets replicated and affect the cells of the host, and what are the possible ways in which this virus can be stopped at various steps. So this is the Chikungunya virus, or it is known as CHIKV virus. So with the help of receptors, it is able to attach to the cell surface membrane of the cells, and then with the help of endocytosis, this virus will be able to come inside the cells or in the cytoplasm, and then membrane fusion will occur. So that will, because of the acidic condition within the uh, near the cell membrane, membrane fusion will occur, and the virus will come out of the membrane, and along with the capsid. So after this capsid is completely out of this membrane fusion, the capsid disassembly will occur, that will lead to the genetic material that is positive single strand RNA to come in contact with to come uh, to expose this genetic material get exposed to the translation machinery of the cells. From this, the 49 RNA genome will translated, and it will then translated into a polyprotein, and within this polyprotein, the NSP non-structural protein from 1 to 4 contains a viral replicase. So after the, we can say, after the cleavage by NSP2, that is non-structural protein 2, this NSP1 to 4 can form a viral replicase, and this viral replicase can then helps in the negative strands template, or we can say replication of negative strands RNA template from this 49S RNA genome, and also that will helps in the transformation from negative strand RNA to 26S subgenomic RNA that can form the structural protein, and which can then come together and form a virus after cleavage after cleavage of this individual structural protein. And simultaneously the capsid formation translation will also occur, and this capsid translation along with the translation from 49 S RNA genome will form together a nucleocapsid assembly. And simultaneously what will happen, this the structural protein will go through this nucleus, and then endoplasmic reticulum Golgi for further post-translation modification to get a glycan structure on the virus. What will happen is that viral glycoprotein will then package inside this nucleocapsid assembly, and then burst out of the cell membrane through exocytosis and form a mature virion, which contains an envelope in the form of cell membrane. Now this mature virion can infect other cells uh, as similarly as it has infected these cells. So, and the things, sorry about it, and the things that are in red is mentioned about the possibilities at this step to stop the infection of chikungunya or replication of chikungunya within the cells. That is, replication can be stopped. For example, in this case, replication can be stopped if the receptors of these cells are been taken over by the some monoclonal antibodies, so that no receptors are available to interact with chikungunya virus, so they will not infect the cells. Another thing that can be done is that use of chloroquine within the cells will prevent the membrane fusion, will prevent the uh, dissolution or membrane fusion or dissolution of the membrane, so that these viral particles or capsid along with the viral genetic material will not come and expose to the cytosols. And this 49S RNA can be inhibited by capping inhibitor, so that this translation will not be uh, happen. And this some silencing RNA can also be used to prevent the translation of this non-structural protein. And uh, if this is formed, then protease NSP2 protease inhibitor, if this is non-functional or somehow this NSP2 can be inhibited, then the cleavage of this non-structural protein will not occur, so the maturation of the non-structural protein will not proceed further. If and another step where this chikungunya virus can be stopped is during replication. So the use of favipiravir or uh, ribavirin. We have discussed the use of these things during our uh, COVID videos, where we are discussing uh, different ways to inhibit the COVID replication. You can check that video. And another thing, next thing that can inhibit the chikungunya virus formation is to prevent the cleavage at the final stage by using DEC, RVKR, CMK. So if there is no cleavage, then the inactive virus will get out, or virus will not get packaged inside the nucleocapsid assembly. So these are some of the ways by which chikungunya virus can be inhibited from replication or infect the cells. I hope you have understood something about chikungunya virus and some some ways by which this virus cycle can be inhibited. If you need to understand about this thing in detail, you can go through the description that has been you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.